Hi, good morning, everyone. Welcome to VICC seminar series for Tuesday. It's my pleasure to welcome and introduce Dr. Somak Roy, who is going to present his work today. I'm looking forward to hear. He's a longtime friend and also colleague, and I'm proud of his work. He's currently an associate professor and director of molecular and genomic pathology services at Cincinnati Children's Hospital. His work focuses on developing molecular diagnostic testing for pediatric cancer using a next generation sequencing. We look forward to your presentation, Somak. Welcome to VICC.
Thank you very much, Samanthi, and thank you, team, for giving me the opportunity to share the work we are doing here at Cincinnati Children's. So I will go ahead and share my screen. If you all can see the presenter mode. You see my slides not in present.
Yep, okay. Looks good.
All right. So I have a lot of ground to cover, and I'll try to be brief, but also balance between the detail of my content, plus to make sure that there's enough time at the end for questions. So the work here obviously is representing a lot of the effort that's been put in the Department of Pathology at Cincinnati Children's Hospital. I serve here as the director of molecular pathology, and it's the result of this is a fruitful collaboration between the Department of Pathology and the clinical team here, especially with the oncology service. So we'll go over a very core of what our laboratory services look like and what the goals are. And then we'll dive into the technology, particularly focused on sequencing technology. That's kind of bulk of our testing relies on. And then I'll summarize sort of the clinical validation of how we develop the test, share some examples of clinical cases, and then just talk a little bit about what we're thinking down the road in the next few years. So the Molecular and Genomic Pathology Services is the clinical molecular diagnostic lab at Cincinnati Children's. We provide high throughput clinical molecular testing for a range of different diseases. This talk is going to be focused on pediatric cancer. The emphasis is on high quality patient care, and we really emphasize on the fact that we are able to develop an effective communication and consultation route as we work with the clinicians in our institution as well as from the other children's hospital. So in the context of development of these tests, it's centered around sort of the whole idea of how we think about pediatric cancer as compared to cancer more in the adult realm. And when we talk about pediatric, really what we're talking about is a range. When we are looking at it's not only pediatric as in its true sense, but also adolescent and young adults. And as we're seeing in the more sort of in the past decade, we've seen a slight increase in the incidence of these different types. Part of it has been improved diagnostics as well. And when we develop or when we approach in developing all of these testing, the idea was to look at the entire picture in terms of the time at which the diagnosis is established via a combination of imaging and pathology diagnostics, as well as what does the scenario look like when the patient is on treatment and the instance that there is recurrence or relapse of the disease. And of course, when we are dealing with pediatric cancer, there is a very important component of germline testing that comes into the picture. Of course, our lab does not do that formally, but we do encounter not infrequently do encounter germline variants that might show up as part of tumor only testing. And so we collaborate with our genetic counselors and the germline genetics laboratory to be able to coordinate more appropriate care once such information is found. Okay, so coming to what tests we offer from an oncology standpoint in our lab. So we have two buckets, one for comprehensive and diagnostic molecular tests. The other focus on minimal residual disease monitoring. And there's a lot of work that we are doing in the lab right now to bring in some more innovative testing. So from the diagnostic perspective, our flagship test is the comprehensive targeted panel. We call it CINCISEQ, short for Cincinnati Children's Cancer Sequencing. And the other test that we do, which is really focused on the FLT3 ITD variant, using a rapid turnaround time PCR, where we're able to turn around the results in 24 hours, primarily from a treatment question perspective. From an MRD perspective, we have a test that we developed a couple of years ago. It's called HistioTrack. This is a digital droplet PCR test that is specifically looking for BRAF V600 mutation, with the intent to monitor the disease level in kids who are on treatment for histiocytosis. And it's the way it has been designed. It's an ultra high sensitivity testing to be able to identify 1 in 50,000 circulating cells that harbor that mutation. Again, the idea is a fast turnaround time test, and the fact that we are able to detect very low level, and hopefully as we go through this process, we're able to help guide our clinicians manage follow-up and treatment of these patients with histiocytosis. So in terms of how the test order workflow looks like in our lab or in our institution, it starts with the ordering provider that initiates the testing. It's typically a process. It's a little more complex than that, and usually for the workflow that looks like for our hematopoietic neoplasms versus solid tumors is slightly different. So for the hem-onc side, the testing typically is initiated by an order in Epic from our providers. Usually it is accompanied by either a blood or a bone marrow. We have the bone marrow clinic that's involved in terms of the actual sample collection and triage. From a solid tumor perspective, every specimen goes through a routine anatomic pathology review of resections or biopsy, and there's usually a discussion and communication that tends to happen between our pathologists and the ordering provider or the clinical oncology team to either recommend sequencing test, or depending on what the diagnostic context is. And in our scenarios, the majority of the indications for testing fall into either a diagnostic question to be able to formalize a diagnosis or establish a diagnosis, and the other one, of course, is treatment options. But interestingly enough, at least in our setting in the pediatric tumors, a good bulk is a diagnostic question in terms of being able to identify genetic alterations that would either bolster or formalize a histologic diagnosis. So in terms of the NGS testing workflow, this is a very high-level summary of what the different steps are involved. There's a lot of focus that we emphasize at the time of tissue triage, with especially with solid tumors, where a formal H&E review and microdissection steps are involved, typically by a molecular pathologist. And we often consult with our anatomic pathology colleagues to make sure that we're able to get the right tumor and the right proportion of tumor cells to minimize our false negative results. And then correspondingly for hem-onc test, we start with peripheral blood or bone marrow, followed by a DNA isolation. We then go through an NGS library prep process, which I'll discuss in a little bit detail in the next couple of slides, followed by target enrichment and sequencing. We then have the data go through an extensive bioinformatics pipeline and a bunch of genomic databases for variant annotation, followed by standard laboratory QC, variant triage, and literature review by a team of variant scientists, and then followed by medical interpretation by one of our molecular pathologists. And then finally, we release a report back into the EMR. Typically, this is in conjunction correlation with our the primary tissue diagnosis, as well as other clinical findings. We do often end up correlating our findings with the findings from the cytogenetics lab. So overall, we get a comprehensive profiling of the tumor. This is a little bit detail into how we do things slightly differently for our rapid turnaround time heme NGS testing. It still uses the same backbone, but in addition, we perform the FLT3 ITD PCR, as I mentioned before, as a rapid component of the test. So typically within 24 hours, we're able to release that information about the same time as sequencing is going on. And so whenever we have heme as part of the sample cohort that we sequence, we usually have a bioinformatics pipeline optimized to be able to get the results, typically within about 4 to 6 hours. And so the full NGS report, or at least the prelim followed by a full report, usually follows in about 4 to 6 days. Sometimes if it is a more challenging case or we need to do some repeat, then it might extend a little bit more. A quick look into how we do the NGS library prep. So for both DNA and RNA, we use the XGen kit that is provided by IDT. For the XGen kit that we use for DNA, it's a single library prep kit that works for both FFP samples as well as for cfDNA, as in including the UMI barcodes in here. For something like a diagnostic testing performed on tissue, we do not leverage the UMIs, but as we're developing more tests for liquid biopsies, that's where we leverage the UMIs as part of the library. On the RNA side, it's what is used is known as an adaptive technology. It goes through what it really helps us in terms of being able to handle really poor and degraded FFP tissue. And especially if these are very small in quantity. And I'll discuss a little bit about the details of how low we can go in terms of the input tissue and the input DNA. So the next thing that I want to focus on is the target enrichment part, and this is typically one of the arduous tasks that is involved when a new NGS panel has been developed. So when I came here in Cincinnati Children's, the idea was to be able to, bring an oncology service from scratch. And so the idea there is to be able to make sure that we are comprehensively addressing, the different types of pediatric tumor that at least, our institution encounters. And really, the ultimate goal is to ensure that, whoever child comes in here with a diagnosis of cancer, that they have access to the test. We don't want to exclude saying, this particular testing is only available for such tumors and, other tumors don't qualify. So the goal was to be able to be as comprehensive as possible and be inclusive of all types of, tumor diagnosis. So really what was involved in the process is extensive literature review, obviously that included the WHO Blue Book classification systems that we went through. But we also had a very valuable feedback from the different subspecialties within our clinical oncology team to be able to come up with that list of genes for RNA fusion-based alteration, structural variants, and DNA alterations. And the way we ended up deciding the design was to go with the hybrid capture methodology, but using a probe set that was part of the GOAL consortium. So briefly, the GOAL consortium is the Genomics Organization of Academic Laboratories. This is a consortium that was started a few years back with the idea of being able to harmonize and develop a collective network of academic institutions, especially molecular pathology, to be able to leverage building these sort of large-scale NGS panels at a relatively low cost and with a lot of flexibility to be able to design our own panels with DNA and RNA. And really, the idea was to be able to keep the cost down so that at the end, the tests that we are able to offer to the patients don't end up being overly burdensome for them. The advantage for our lab has been we've been able to tap into the expertise across more than 35 institutions in the GOAL network from a range of topics, including assay setup, bioinformatics, clinical knowledge base, and sample exchange and validation. So what does our panel look like? We did have to customize a little bit of additional genes because the initial GOAL probe was designed more from the adult sequencing perspective. So at the end, we ended up having about 388 genes for DNA mutations and tumor mutation burden calculation. We use about 531 genes for CNV LOH analysis. For gene fusions, we designed the same probes that were meant for DNA with about 184 driver genes in mind. The way the fusion probes were designed as part of the assay was the idea that it could not only pick up known partners, but any novel partners as well that we do not sequence as part of our panel. In terms of sequencing, we use the Illumina sequencers. It's a fairly standard technology. I don't want to get into details of this. This is how our bioinformatics pipeline looks like. So we start off with the raw sequencing data coming off of the sequencer as BCL files. The two flagship instruments we have is the NextSeq 2000 and the NextSeq 550. So once the FASTQ files are generated on our system, it then forks into the RNA and the DNA pipeline. The DNA pipeline typically involves the standard types of pre-alignment and QC, followed by sequence alignment using BWM2 and post-alignment QC steps. For the sequence alignment, both for DNA and RNA, we're on the GRCH38 version of the human genome. Followed by the post-alignment QC, it then splits up into copy number calling pipeline and variant identification. For variant identification, we utilize two different algorithms. Vardict is used as a standard variant caller, but also as an enhanced indel detection tool. And then for FLT3-ITD, given its complexity, we use a dedicated caller for that to make sure that we minimize our false negatives there. That is then followed by aggregation of the variant calls, and then we annotate that and prioritize it. We use a combination of different tools using an IGVS Python package for the annotations, ECVAR for the annotations across different databases, and we have some custom apps that we've created around that to streamline the output and get it ready for variant interpretation review. So this is where our DNA variants and the TMA information comes off. From a copy number standpoint, we use CNV-Kit. That is kind of our standard tool for generating the panel of normals because we do not do a paired tumor normal sequencing. And that is then used as a benchmark to then generate the copy number calls for each of the specimen. We developed a custom tool for visualization of the copy number plots. It's called CNA Plotter. That's available freely available on GitHub. And we use that output then to do the visualizations. The interpretations of our copy number calls are predominantly based on visualizations rather than numerical cutoffs, just given the fact that as we see a variability in how these copy number calls appear, depending on the amount of tumor tissue we have from the tissue sample. Switching gears to the RNA side, for RNA, we go through the pre-alignment and QC process using FASTP, followed by alignment to the reference genome using star aligner. Following that, we have two sets of workflow. One of them focuses on the more standard fusion calling and SV detection algorithm using chimeric read count and fusion prioritization. For that, we use two algorithms. One of them is star fusion, and the other one is Ariba. And then that ends up giving us our gene fusion calls and the structural variant calls. At the same time, there was a requirement from a clinical lab testing standpoint to be able to identify exon skipping for specific genes that are clinically relevant. And notably, these are EGFR variant 3, MET exon skipping 14, and beta-catenin. And I'll give you an example of how we use beta-catenin for pediatric tumors in terms of being able to use exon skipping to detect large variants. So that kind of summarizes what the pipeline looks like. The infrastructure that supports all the pipeline and the global workflow of the lab is a work in progress. Right now, we are in the process of deploying our custom lab information system, which in the phase 1 and 2 we've been able to interoperate with Epic and Beaker systems in being able to electronically receive the test orders and patient information. This also integrates with how we onboard these specimens and record and perform instructions. There's work in progress in terms of being to develop automation for some of the more wet lab steps. From the bioinformatics pipeline side, most of our stack that we develop and maintain is within Python, C++, and Go, where a lot of the custom algorithms and scripts that we maintain to sort of optimize some of the QC results or the integration of the analysis to generate reports for the clinical review and reporting. Overall, at a very low level in terms of how we have deployed the entire pipeline and the apps, we use containers from development to production through the entire life cycle. And these are deployed on our Kubernetes cluster. For CI/CD, we use GitHub for code management and automation. So coming to variant interpretation, when all of these results are ready for review, we go through a process that I'm sure many of you here are aware of, starting from the variants from the VCF, going through steps of normalization, generation of the HGVS nomenclature, getting the right gene names, and the other metadata from using the annotation tools, and then finally using a combination of those data points for interpretation. We use two sets of guidelines to be able to help us do that. One is the variant interpretation guidelines that was released by AMP in collaboration with ASCO and CAP that triages the variants into 4 tiers. And this is what we use for clinical interpretation when we ultimately put in these in our clinical report. And again, I'm not getting into details here, but the two extremes are, the benign variants or likely benign variants, and then tier 1 and tier 2 are for variants of strong clinical significance and potential clinical significance. Specific to our institution's requirement and to keep things relatively simple in our report, we report variants under these two tiers 1 and 2 as a combined group. And then we separate out tier 3, which is VUS, and then tier 4, which is benign, for sort of practical operation purposes. Now, before we go to the variant interpretation guidelines, as we use when we start to add pathology and clinical data to it, we use the variant interpretation guidelines or the oncogenetic SOP. That sort of helps us triage variants that we have never seen before, to go through a systematic process of how we prioritize those in terms of whether they are benign or whether they're oncogenic, kind of from a biological perspective. And then we apply the clinical information that we have from our pathology reports, from immunohistochemical findings, from flow cytometry, cytogenetics, and then the clinical features. In terms of databases, this is a long list I'm going to go through in details, but it's a standard set of databases that we leverage for cancer variant annotation. Of course, as I said before, it is not uncommon to find incidental, suspected or potential germline variants. So we do tap into some of the variant databases that are useful for germline as well, and that helps sort of triage the variant interpretation for our lab. This is a standard that I think most lab use in terms of mutation nomenclature. So we use the HGVS recommendations for that. This is a very, very simplified and sort of, abridged version of how variant sort of, tiering happens in our lab. And again, a lot of the things have been sort of simplified in terms of being able to summarize the process, but the actual, details are not much more, elaborate in terms of how each of these steps happen. So we typically, when we start off with, we split the workflow based on if you're dealing with loss of function genes or tumor suppressors versus oncogenes or gain of function genes. And we kind of go through the process where we first look at the type of variant based on the genomic position, the HGVS nomenclature, and the location of the variant, whether it is in the coding region or in the non-coding, for example, intronic or the UTR regions. We then go through a filter of population frequencies, depending on what the cutoff is. This is a little old slide that says a 1% cutoff. We actually follow a 0.1% MAF cutoff. Again, whether it is global frequencies or ethnicity-specific ones. What is important here in our experience is when we start to see frequencies that go higher than 1% in specific ethnicity or populations, we are a little bit careful before we automatically classify that to a benign variant, just because of founder effect of certain pathogenic variants. And so that's something that we keep in mind before we. So it's typically not an automated process for when we encounter only 1 or 2 ethnicity specific populations that kind of, suddenly jump up in terms of their allele frequencies. Followed by that, once it goes to that filter, we then look at the variant frequency. First we look at for truncating mutations, we look for to see if the location of truncating variants, if it is upstream of the 50 nucleotides from the last splice junction. For missense and inframe variants, we look for variant prevalence and occurrence of hotspots, whether it is cancer specific or more global across different cancers. For the intronic or splice site variants, we utilize the in silico algorithm predictions for splice sites that is taken into account. And ultimately, each of these findings then go through step of literature review or referencing our own internal knowledge base to be able to, which is typically done by our variant triage team and the molecular pathologist, before we come to a final conclusion as to how we classify these variants, ultimately, which is tracked by our system in terms of the in-house knowledge base. We do have a provision here where when VUS are called, sometimes we flag a VUS, and typically the flagging here means that we don't have anything else to bank upon in terms of calling it, pushing it to a tier 1 or a tier 2. But there are subtle pathology findings or clinical findings or some other, say cytogenetics findings that raise a suspicion. So we ultimately have those flags being sent over to our final molecular pathologists when they review it, so that they can make that final decision. So we tend to take VUSs which sort of hover at that borderline of tier 2 versus VUS a little bit more cautiously before we kind of label it with a certain classification. This is gain of function. So this is again sort of the same thing, but in terms of the steps, but a lot of it is heavily driven by the variant type in missense or inframe, and a lot of emphasis and calculation goes behind calculating hotspots and frequencies across different domains of the key protein regions, for example, tyrosine kinase domains or the juxtamembrane domains for certain tumor type. We also there's an extensive literature review that is involved, because as most of us know, trying to interpret gain of function variants is typically more challenging than than tumor suppressors. But we have a very it's the literature review process is a little more involved typically when it comes to oncogene, at least in our experience. But again, it's the same process as we go through the triage, the variant triage process, and at the end we end up documenting a variant, whatever has been classified. We keep track of that through our clinical testing, and depending on when new information is available, if we encounter that variant again for a different clinical case, then we spend time to see if that requires a reclassification or it remains at the same class. Touching upon a little bit in terms of how we did our validations. So again, this is a test that went through the analytical validation process for high complexity LDT molecular testing in compliance with CAP and the CLIA regulations, and of course following the guidelines that's are available out there. A quick summary of what the clinical cohort looked like. So it was done as a methods-based validation. We had a total of 120 different tumor samples, of which 112 were clinical, and we used 8 reference materials to augment the number of variants. From the reference material, we got an additional 4,500 variants, and it was split up between the different sample types. So about 60% of them were tissue or FFP samples, and about 38% were peripheral blood and bone marrow. In terms of the tumor types, we tried to cover a large spectrum of different pediatric tumors, including brain, soft tissue, histiocytosis, liver were kind of like the predominant ones. And in our cohort, and of course from the heme side was ALL, AML, and lymphomas. From the types of variant, we obviously had, as you can see, the bulk of it was SNVs. We carefully picked the SNVs as part of our validation cohort to also represent areas that are difficult to sequence. For example, the CBPA gene and the TERT promoter region, which are highly GC rich, and they tend to often cause a lot of noisy sequence. And so we made sure that we had good representation of those difficult, challenging SNVs in those regions as well. In addition, we had a combination of short and long indels. We had an arbitrary cutoff of 10 base pairs as that because of how we used or how we tuned our variant caller to be able to pick those up. We had about 103 CNVs and then 72 fusions and structure variants. So overall, we had a positive percentage agreement of 99% for SNVs, close to 100% for short and long indels and fusions. And then for CNV, it was about 98.1%. This was partly limited due to some of the samples that were at a tumor cellularity below 50%, where we see the CNV algorithm struggle or decrease in sensitivity. In terms of limits of detection for analytical sensitivity, from a SNV, MNV, and small indel perspective, there's between 3 to 5% VF at a minimum of 300x coverage. For long indels, we are at 5 to 10% VF. We do see sometimes we are able to pick up slightly below that. And for fusion, we look for 5 spanning reads across the fusion breakpoint from both the algorithms. In terms of percentage tumor cells, particularly for SNVs, MNVs, and indels, we can go down to as low as about 6 to 20%. We try to keep it 50% or higher whenever possible, just to be able to report out the CNVs. But in the instance that we don't have anything available, CNVs can be challenging, but we are still able to pick up key driver alterations in at least from SNV and indel perspective. From a positive predictive value perspective for DNA, CNVs, and fusion, we were above 98%. So I'm going to go through a few clinical case examples to highlight some of the advantages of the way we designed the panel and how we can handle really small tissues. So to begin with, this is an example of a 22-year-old male with an atypical meningioma diagnosed on histological impression. The tissue size you can see is extremely small. These are like little pieces of the size of a little nail, if you can imagine that. Of course, not surprisingly, the DNA and RNA quantity were pretty low. But we were able to successfully pick up the fusion here. This is a YAP1 and MAML2 fusion. This has been described typically in the cases of pediatric meningiomas or young adult meningiomas where these are NF2 wild type. And this is a relatively new molecular entity that requires the identification of this particular fusion to make the diagnosis. The next case here is an example of another brain tumor. It's a 15-year-old male with a polycystic astrocytoma, again, dealing with very small tissue and low input DNA and RNA. Here again, we were able to pick up this characteristic fusion described in polycystic astrocytomas, which involves the KIA1549 and BRAF fusion. You can see here the number of reads are almost bordering to where our limits of detection are. And this is not only because of the small size of tissue we had, but also because of the challenging nature of this particular fusion. It's and again, this fusion is inherently difficult to pick up on NGS-based RNA fusion panel, but we've been able to optimize how we design the probes and of course the bioinformatics to rescue some of these low-level calls that we see with this particular BRAF fusion. This last case is an example of a liver mass, which on histopathology was diagnosed as a well-differentiated hepatocellular carcinoma rising in a hepatoblastoma. And this is an example of where we use the exon skipping to our advantage. So you can see here on the top panel shows the IGV snapshot of the sequence read in the beta-catenin gene. So pediatric liver tumors are known to have these alterations in the beta-catenin genes that involve large deletions. So the canonical beta-catenin activation mechanisms are SNVs and small deletions in exon 3 and sometimes in exon 7. But with certain subset of pediatric liver tumors, they tend to have these very large deletions that involve exon 3 to the point that the entire exon 3 is knocked out. And as you can see here in this example, this is a 557 base pair deletion. It's quite large, and given the density of the probes here is how we were able to pick this up on our DNA panel. But during validation and prospectively, we've seen that it is very challenging, and sometimes our DNA panel is not able to identify this large deletion. Sometimes is due to the fact that it is very large, that it involves multiple exons, and there's a lack, then the alignment becomes very difficult. In other times, you've seen them being called in a way where it's extremely strand biased, and so the variant caller would either flag it or not call that. And so what we do is we use the exon skipping algorithm that we developed in-house to be able to rescue these calls where we can actually clearly see the exon 3 either partially or completely lost. And so we use that to be able to rescue such cases where the large deletions are not called by our DNA panel. So in this case, of course, and it was called both ways. So it's a nice correlate of what we see in the DNA sequence and then the RNA sequence here. This tumor, in addition to the beta-catenin, also had a TERT promoter mutation that is consistent with the rendered diagnosis. So overall, in our experience since January of 23, when we launched the test, we've done over 400 pediatric tumors with a failure rate of less than 2%. And most of the failure rates have been associated with severe FFP artifacts and RNA degradation. Sometimes these are really old blocks that are older than 5 years, and often that's difficult to sequence those. Sometimes it is due to excessive hormone exposure. On an average, we are able to generate about 1200x coverage across different samples, and our mutation load detection, as I mentioned before, we range between 3 to 9%, and we are able to consistently pick up variants at as low as 3%, especially for histiocytic neoplasms, post-treatment tumor necrosis, and vascular tumors. So where are we going forward from here? And I'm sure this is something that is a common theme across many molecular labs who are involved with pediatric and young adult tumors. We are trying to expand the cohort from these more focused testing as we're doing right now into getting into integrated viral sequencing testing, introducing methylation as part of standard of care, which already has been done for brain tumors. So our lab is trying to bring that up as part of the comprehensive offering. And again, as I mentioned before, there's a lot of work that we're doing in terms of MRD in bringing up tests. So HistioTrack is one of our flagship histocytosis tests that we used for beta V600E mutated histiocytic tumors that we monitor them. And we're trying to expand that out to other MAP kinase pathway mutations that would then help that test become available for other patients, who have more systemic, histiocytic lesions that tend to be negative for beta V600. And we are also expanding our efforts into developing liquid biopsies for monitoring patients with solid tumors. One of the projects that we have piloted and we're looking into the data is for pediatric liver tumors. And so with that, I want to acknowledge, our team here in pathology, all the pathologists that have been exceedingly helpful in terms of collaborating and developing the test, the histology lab, obviously all the members of the molecular pathology lab, and our clinical colleagues from the CVDR team. And thank you for your time. And if there is any time, I can take some questions.
Thank you so much for the great overview of all the tests, which is very important, especially the variant interpretation you went through in detail. I have one question about like a very basic question, and then I will open if anybody is interested to like discuss. I just saw one very basic question I have is like, when you say like you only do the tumor sequencing, what kind of normal samples do you use? Is it the pediatric normal samples? Can you, like how, because the TMB recent papers have shown that TMB is biased if you do not have a matched normal. So how do you address that question?
Yes, that's a great question, and it's challenging in the sense that ideally if we had from a financial standpoint, it's difficult to be able to sustain sort of paired tumor normal, but doing tumor only sequencing, as I mentioned before, we had sequenced a cohort of normals when we were doing the validation, and that is that's a key thing that we do for a lot of different modalities, including the fact that TMB can be biased. So the panel of normals initial. So what we typically do is the source of the specimens. Since majority of our samples are in the pediatric setting, we do try to keep it that way, where these, even if we, for example, we are taking samples from normal intestines that are resected as part of a non-neoplastic disorder, or tonsillectomy specimens that come out where they've been reviewed and been shown not to have any diagnosis of tumor. Whenever we are picking up these samples, we tend to make sure that they are age represented in the 0 to 20-25 years range, which typically is what we get in our anatomic pathology lab. So yes, our panel of normals are largely represented by different solid organs and peripheral blood or bone marrow samples that encompass that age range. But of course, after a careful review, we make sure that there's no significant oncology history in these samples that we're looking at.
Thank you so much. If open to the question and discussion.
Yeah, I'll ask a quick question. Great presentation.
Thank you.
So you talked about doing some normalization. What normalization algorithms are you using in your bioinformatics pipeline post your, like your VCF results?
Yes. So let me see if I can go back to our slide there. Okay. So the normalization step that we involve, so we use the BCF tools, the norm functionality of the function that it provides. Typically, what we're doing there is as soon as we have the raw variant calls from Vardate or from the FLT3 ITD, we then pass them through this normalization algorithm, and it does two things. One, it is, and this has been described in the manuscript by the authors of VT application. We used to use VT before, but then we switched to BCF tool norms for just ease of use purposes. But what it is doing is it left lines the variant so that on the genomic coordinates the variant is represented at the most as the leftmost variant, and this is particularly with indels that occur in short or slightly longer repeat regions, whether it is mono, di, or trinucleotides. The second thing it does is it tries to represent the variant as the smallest possible way of what a variant can be. It generally works pretty well in terms of downstream annotation, so we don't end up misannotating certain variants. We do encounter a little bit of trouble with sort of complex variants that are del-ins, where it's a variant that is called because there is phased information where there is deletions or insertion or SNV and a deletion on the same reads, and they end up getting called like that. The algorithm still tries to normalize as best as possible, but we do a manual review to make sure that we are getting the correct AGVS nomenclature for that. But yeah, our go-to tool is BCF tools.
Thank you.
So,
yep.
Dr. Roy, it was a great presentation. I have one question.
As
we are dealing with the pediatric population, and you often also get sometimes like germline variants. So I was trying to understand in that your variant prioritization tool, if you find something which is common in population, but still you find it has been reported in cosmic or somewhere in a somatic state, how you are dealing with those kind of variant?
Yeah, that's a great question. And I don't have a black and white answer, but I'll try to at least describe what we do. So, in the context that we identify these potential, pathogenic germline variants that come across our pipeline, of course, the general rough rule, which I also use it with a big pinch of salt, is anything around 50% VAF. Yes, you can use that as a guidance, but that generally, you have to interpret with caution, particularly because in pediatric tumors, it is fairly common to get tumor samples which are wall-to-wall tumors. So, small round cell tumors, whether it is in the kidney, liver, brain, soft tissue, they tend to be very rich. So you might have driver mutations at about, close to 50%, but you would also have potential pathogenic variants that are also, germline variants that are also at that level. So that's not very helpful to distinguish that way. But what we do is we tend to use that as an initial flag, but not jumping on saying, oh, this is a germline variant. But then we go through the process of reviewing the variants to see what is available or how it has been described in the public databases, in the published literature. When it comes to the point where we're looking at, okay, how many times has this been described in Cosmic, or how many times it has been described in, ClinVar or, DBSNP, it is important to understand that there is always cross-contamination across these databases in terms of how they've been, have been marked. And I know in ClinVar, there's a lot of effort going on to, really standardize the curation, which is very helpful, but still it is to be interpreted with caution. So what I mean by that is, for example, if I see a variant that ends up being in Cosmic, I don't know, 5 times or 6 times across maybe 2 tumors, that by itself doesn't mean that it is a cancer-associated variant. Similarly, if you see a variant that has been, documented in ClinVar or DBSNP, maybe 4 or 5 times with or without a clear context, it does not automatically mean that you're dealing with a germline variant. So, in those instances, when we see these more rare variants that, we don't have much information about from the public database, but for example, they're falling in the right, hotspot domain where other pathogenic germline variants have been described. So those do require obviously a very detailed review, but we exercise caution. We also obviously go back to the patient's chart for any prior testing. That is often very helpful, because when patients get, they transfer from another hospital system to ours, we do see a good subset of them, having had some germline testing before that identifies that. But obviously, if we don't have that information, again, we then, work with our oncologist and the genetic counselors to be able to then route them through appropriate further review. I mean, since we mostly work in the somatic space, we don't go and do an extensive review of the germline variants, but we do flag them and have, alert our clinical team and the genetic counseling team to be able to, further take it up if based on their clinical interpretation, if they feel the necessity to go through a full medical genetics workup. But that's typically how we triage that. But it's not, as I said, it's not a black and white scenario. We do end up having challenging scenes, but the good thing is we do have a fairly big genetics team here that really gets into detail of many of these findings.
Thank you.
Sure.
I'll go one more time. I was really happy to see that you guys are using the oncogenicity guidelines, at least in some level of prioritization.
Yes.
I'm at Nationwide Children's. We're in the process actually right now of preparing our own internal SOP for using, these guidelines and trying to clarify and then validate these. Have you kind of gone through, have you done like an official validation and tweaking of these so that you can have like the level of clinical rigor that's really required? Or where do you kind of like, where are you in this process?
So, in terms of the analytical validation, we have gone through significant validation in terms of how we are implementing these guidelines as part of our workflow on purpose. Well, since we know that except for the example of say population frequencies where you can really to a certain extent automate how we end up flagging them as potentially benign. Of course, you're trying to get scores which are in the -6 to -8. But beyond that, the interpretive process is very involved, and there are a lot of edge cases. And so the path that we took was, the whole guideline implementation was part of the validation, but we have kept a lot of wiggle room for review and interpretation with our variant triage team and our pathologist. So yes, we tend to flag things, but we don't put hard filters. So for example, if we have, say, 800 variants that are called in a given sample, we would flag them appropriately as, if certain things are meeting the very strong criteria for benign, we would flag them as benign. But we do not hard filter them out so that our team doesn't see. Our team gets to see every variant that was caught and whatever flag was in there. So what this has helped us is, as we have been kind of going through, since we have launched it, we've been able to identify certain trends as to where we can maybe think about automating a little bit, certain aspects of these guidelines, whether it is coming from AMP or from the oncogenesis SOP. And so we've been incrementally adding those automation steps in terms of flagging so that it makes our life a little bit easy. But that obviously gets incorporated into our automated workflow after we have done the validation of those. But to a large extent, the variant interpretation part is still quite extensively reviewed by people after appropriate prioritization. So that's where we have left it at.
Yeah, absolutely right. And I think there's, I'm in a couple different groups, like, very interested in talking about this. And the things that we're really considering here locally is like, what kind of criteria are needed for, the terms like well established for your functional studies, or,
Right.
All of these little things that there's enough nuance that like you're gonna always basically have to have some level of human interaction in some of these. But then making sure that you can enable consistency is something that we're, really trying to focus on here, and trying to, iron out some of the little details so that, each of our variant scientists, are being consistent with the way that they're applying the codes.
Yep, that makes sense. And again, one common, and this is, based on our experience with pediatric population, well-established functional studies are, it's like it's a dessert after your meal, which you often don't get to. There are not large studies. There are, unfortunately, fortunately, since pediatric cancer is rare, unfortunately, there's not as, detailed of a study as you would get, say, for example, if you compare to lung cancer or melanoma in adults, it's a much larger cohort. So that is the one thing which I think, as a community, we need to have to work on and refining how to deal with those things when we talk about, do you have sufficient functional evidence to say that, yes, this is likely oncogenic or not. And again, in the context of pediatric tumors, which tend to be super rare in some instances.
I think we can take one more question. It's like 3 minutes. So there, Beth, you want to go?
Great, thank you. Thanks so much for the presentation. I found it very interesting and actually kind of validating to a lot of the development that we've been doing at Mayo Clinic. Yeah, so it's been really great. My question is about your copy number plots. We recently went live with kind of our own custom copy number plot as well.
Okay.
Looking at NGS data, and we're using both on-target and off-target reads with a very similar schema. So I was wondering a little bit about like, and we also used a panel of normals. I think we had like 50 of each. So yeah, it looks very similar. Are you able to, so are these just your on-target reads, like your probes from your chemistry, or are you using supplemental data here too?
Yes, that's a really good question. So whatever data you're seeing here, so this is the algorithm that we use to visualize the data that is coming from CNV-Kit. So CNV-Kit, I don't know if you're aware of it or not. It tends to obviously tends to let you demarcate areas based on your panel of normals or whatever other criteria you specify, where you can flag regions of your genome that is kind of within your scope of sequencing as anti-targets versus targets. So we try to use that and only focus on the targets. So anything which is on target, good mapping quality, makes it to that cutoff. And then whatever we are using for visualization is only those on-target reads with consistently good coverage. We do have some, certain areas that tend to drop out, like for example, the SAMD9 and the SAMD9L gene as examples. They are always slightly lower than every other gene, but it's not so low that we would end up calling deletion of those two genes every single time. So we've been able to kind of adapt to that a little bit, so we don't end up discarding the genes altogether. But other than that, we tend to really only focus on the on-target rates. What we are in the process right now is we are actually finishing up a validation for augmenting our copy numbers, and that is including an LOH copy number backbone probe. So what we are doing is we're supplementing our current gold probes with a backbone probe from IDT. And so that's essentially probes that are sequencing a bunch of SNPs across the intergenic and the non-coding regions. So that essentially helps with the LOH calls in addition for the copy number calls, to be able to rescue some of these low-level calls that so far we have been having a little bit of a challenge with. But that's in the works. It's not there yet, but that's a supplemental probe to the copy number probes.
Yeah, and that's very parallel to what we've been doing too. So we just went live with a B allele frequency. It's very low resolution B allele frequency that we're deriving from TSO 500 data.
Okay.
But we're live with it, so we can tell like if your entire chromosome 17 is copy neutral LOH and that kind of thing. So that's kind of nice. Cool. Yeah, thank you so much. This was really neat.
Yeah,
Great work.
absolutely. Yep.
Yeah, thank you so much for presenting today and sharing your experience. I think this is a great for our group. Yeah, welcome again, and hopefully you'll be a part of VICC also, and you'll be able to join us in our seminars and conferences in future.
Good.
Thank you all.
Thank you. Thank you for having me. Bye.
Thanks so much, everyone.